that, oh yeah, I understand that. So I implemented some videos to the session, and uh, we will watch them today. Upload videos, and also maybe upload videos YouTube. I'm just noting that what what kind of homework you will have. However, you will have no homework basically, but maybe I will share with you some videos if you will watch them individually. And again, they will not be mandatory. Any questions regarding the structure and topic? No? Can we start? Okay, perfect. Um, Satsuki, please, can you read?
How many molecules of ATP are produced in glycolysis? A. 1, B. 2, C. 3, D. From 13 to 36.
Thank you very much. Thank you very much, Satsuki. I have a few more seconds, and let's do it. Two more people. Okay, so here are the answers. Let's discuss them. Um, Let me show you the slide of glycolysis. Here we have the slide. So, first of all, glycolysis is happening in the cytoplasm, as we know. And the outcomes are two molecules of pyruvate, two molecules of ATP, and two molecules of NADH. Nevertheless, if we watch the picture, we see that we have four molecules as an outcome, or the arrows are going from. So why we do count it as only two molecules of ATP? Because we invested two molecules at the beginning to trigger the reaction, to trigger the process and the pathway uh, initially, and then we have four as an outcome. So total net worth or assets are two molecules of ATP, okay? Because we invested two, so if there will be only two as an outcome, the net worth would be zero. So that's why we count it as two molecules of ATP. So and here we have a pyruvate. As you remember, or we will get there later. So most of you in this case went for absolutely correct information, which is 2. From 30 to 36 or 38, it's another part of cellular respiration. Cellular respiration step, which produce the around 30 to 38 molecules of ATP, 38 in the best scenario, let's say, is the respiratory chain. If you remember yesterday, we highlighted that if the oxygen is involved and we have ATP synthase, this step produce around 30 to 36 or 38, depends, ATPs. Okay, and the respiratory chain is happening in the inner mitochondrial membrane. Those This pathway, also as Krebs cycle and glycolysis, we presented or we covered in a very superficial way. However, in just in order to have an overview of those metabolic steps pathways, you will cover them in more detail in higher years or in during the medicine studies. Nevertheless, it is very helpful already for you to have an overview because some questions like this can also occur in admission. Okay, but very well. In glycolysis, we count that on we have two molecules of ATP produced. Okay, very well. Krebs cycle. Misato, please, can you read?
Yes, life cycle happens in A. Cytoplasm, B. Mitochondrial matrix, C. Inner, my, inner mitochondrial membrane, D. Ribosomes.
Mm-hmm. Very well, thank you. One more person. Okay. Yes, here we are. Here are the results. Very well handled again, guys. So, mitochondrial matrix. Matrix is exactly the place or the space where the Krebs cycle is happening. Krebs cycle is also called citric acid cycle, and for entering this stage of cellular respiration, we need to have acetyl coenzyme A. If I ask you what kind of metabolic pathway we discussed so far is happening in the cytoplasm.
Glycolysis.
Perfect. Great. Glycolysis is happening in the cytoplasm of the cells. Mitochondrial matrix is responsible or is the place where Krebs cycle takes place. And inner mitochondrial membrane, do you remember what metabolic pathway takes place there?
Oxidative phosphorylation.
Perfect. Exactly. Oxidative phosphorylation or also oxidative phosphorylation, or also the synonym term is respiratory chain or electron transport chain. All of those titles are correct, so whichever you want to use, you are able to. I guess oxidative phosphorylation is the most common term for this pathway. Okay, and what's happening on ribosomes, guys?
Protein synthesis.
Perfect. Protein synthesis, especially translation step. And we will finally today uh, get little more in depth regarding translation on ribosomes. Very well. Perfect. Next one. Now, please, can you read?
Yes. Which of the following is the primary function of the respiratory chain? A. Production of glucose. B. Synthesis of proteins. C. Generation of ATP. D. Formation of lipids.
Perfect. Thank you very much. Now, let's have few more seconds. Okay, so here are the results. Great, guys. All of you went for correct option, generation of ATP. So exactly how what we discussed at the beginning of a session. Oxidative phosphorylation produce from 30 to 36 molecules of NTP. So it's pretty huge difference if you compare the ATP production from oxidative phosphorylation to Krebs cycle, where we have only two molecules of ATP, or glycolysis, where we have also only two molecules of ATP. So it's pretty huge difference, okay? So more than 15 times more energy we produce in oxidative phosphorylation than in glycolysis or Krebs cycle, for example. Great. Um, okay, so primary function, we know that primary function of respiratory chain is generation of ATP. Nevertheless, production of glucose we mentioned yesterday. Do you remember the pathway or the name of pathway which is responsible for production of glucose? We used one specific term in translation, or in direct translation we can say it's a new production of glucose. Do you remember this pathway? It was at the end of the session.
Gluconeogenesis.
Perfect, exactly. Here we have it. Gluconeogenesis. Gluco represents or states for glucose. Neo is new, and genesis is production. If you remember, we said that really we can say that gluconeogenesis is just in the opposite direction of glycolysis. So we have two pyruvate molecules at the beginning of gluconeogenesis, and it undergoes the opposite direction of glycolysis in order to get glucose. Okay? So from pyruvates, two molecules of pyruvates, we get one molecule of glucose. That's because pyruvates have three carbons in the formula, you see, and glucose has six carbons. Okay, so uh, 3 plus 3 makes 6. And meanwhile, it has to undergo some enzymatic reactions. So we can say glycolysis and gluconeogenesis are almost the exact opposite reactions. Yeah, glycolysis breaks down the glucose, gluconeogenesis produce glucose or is the result of the reactions. Okay, we do not have to spend much time here. Nevertheless, just for you to know the terms, if something like this or this term you will face in the future. Okay, perfect. How do we call synthesis of proteins in medical terminology? We already mentioned this term today. Synthesis of proteins we call also in one word somehow. Do you remember? You mentioned that already today. We call it proteosynthesis. Proteosynthesis. Okay, let me write that. Proteosynthesis. What's going on? Why it's not here? Okay, proteosynthesis. And which organelle or which organelle has as primary function the proteosynthesis or organelles?
Ribosomes.
Ribosomes, perfect. Ribosomes, and also we can say endoplasmic reticulum plays a very crucial role in it. But the ribosomes, we would go for ribosomes in the first place. Perfect. Okay, what about formation of lipids? Yesterday at the end we mentioned synthesis of fatty acids, basically. All of the metabolism of fatty acids or lipids somehow revolve around this molecule, acetyl coenzyme A, acetyl coenzyme A. And if we have enough acetyl, this here we have molecules of acetyl coenzyme A. If we have 8 molecules of acetyl coenzyme A and we undergo the synthesis of fatty acids, basically they bond together and they create one long 16 carbons long fatty acid. Okay, so this is a synthesis of fatty acid. If we go from here, this direction, from acetyl coenzyme A, we synthesize a fatty acid and we have this long chain of 16 carbon fatty acid. If we start on the other end, on the other direction, we call this process beta oxidation of fatty acids. So basically a long chain of 16 carbon long fatty acids is cut, okay, and break down into acetyl coenzyme A. Why acetyl coenzyme A is important for us? Because acetyl coenzyme A as a molecule can enter the Krebs cycle. And if we have a molecule which can enter a Krebs cycle, it produces besides ATP also NADH and FADH2, and all of those things or the outcomes of citric acid cycle can enter the oxidative phosphorylation. What does it mean in other words? Metabolism of fatty acids can also lead to a oxidative phosphorylation and production of plenty of energy. Okay? Not only from sugar or from glycolysis, also from metabolism of fatty acids or lipids, we can get plenty of energy. Okay? Perfect. Perfect, perfect, perfect. Yes. So let me just highlight these. Great. Um, Sora, please, can you read?
Okay, which cell organ is responsible for energy production in eukaryotic cells? A. Golgi apparatus. B. Mitochondria. C. Endoplasmic reticulum. D. Lysosomes.
Thank you, Sora. Perfect. All of you went correctly for mitochondria. Yes. Mitochondria also are, we call them as powerhouses of the cell. Yeah? Perfect. Um, what about lysosomes? What do we know about lysosomes? What they are responsible for?
Lysosome is noni cell is gathering and digesting and recycling.
Perfect. Yes, so we use this analogy as a trash can, but we like uh, ec- uh, put it in another level, let's say, and we describe it in more medical terminology. So it's responsible degrading and waste disposal, also as for recycling the material, unuseful material. So great. Perfect. Lysosomes. What about endoplasmic reticulum? What kind of endoplasmic reticulum do we have? We have two types basically.
Rough and smooth.
Perfect. Rough and smooth. Rough have on its surface ribosomes. Smooth does not have on its surface ribosomes. Um, what is it responsible for? An endoplasmic reticulum. Do you know its function? What functions we have mentioned?
Endoplasmic reticulum works as a factory
hmm
and it producing some products for cell or another for cell itself or something outside organized.
Perfect. For cell itself or also for other needs in the organism, so for basically outside of the cell. Perfect. We mentioned that endoplasmic reticulum can synthesize proteins, also lipids, but also I think we mentioned just as a side information, it is responsible also for detoxification. Detoxification, basically, guys, you can imagine that, for example, liver cells. We mentioned liver cells, they had a lot of mitochondria inside. And also, they are rich for endoplasmic reticulum. Why is that? Because liver is one of the most important organs regarding metabolisms. Rega- uh, there are plenty of metabolic pathways going on in liver, and besides production of proteins or lipids in the plasmic reticulum, also detoxification is going. What does it mean basically? If you have, for example, higher blood pressure, you take pills, drugs against high blood pressure in order to lower the blood pressure. So you take these pills and there is some drug inside, some substance. Liver will try to metabolize it and get rid of it. Okay? Another example, more unfortunate, when you are drinking alcohol and you have an alcohol use disorder. Alcohol is also metabolized in the liver. If the liver cells are damaged, your organism is not able anymore to function properly because the metabolic pathways are not working properly, and also alcohol cannot be metabolized in your body anymore because liver is not functioning properly. And that's because detoxification takes place mostly in the liver cells. And if we are talking now on the liver, on the cell perspective, it takes place in the endoplasmic reticulum. So that's an additional function of endoplasmic reticulum besides the only production or synthesization. So it's really like a huge factory with more departments. One is producing lipids, another factory is producing proteins, another part of the factory is responsible for detoxification. So it has to maintain and work hard to get rid of the substances which are not common or known in the body. Okay, you will know more about the drugs and then the treatments when you are be studying pharmacology in the fourth year of studies in medicine. Okay, and what about Golgi apparatus? What do we know about Golgi apparatus? I will bring the picture back.
Coach apparatus is like post officer, then modified protein, lipids, carbohydrate.
Perfect. Yes. So exactly. Golgi apparatus. Here we have the picture here. It's very close to endoplasmic reticulum, so it's not responsible for protein synthesis. However, it receives products from endoplasmic reticulum and then it decides where it goes. Very good. Very, very good. Very, very good. So, yes, that was it. Perfect. So that was intro, guys. Yesterday we had more intro quizzes. I guess, or at least I assume we recapitulate a lot of things. So today we will focus more on the core and on the main topic, which is nucleus. So as I said at the beginning of session, firstly, I would like to maybe provide you with more information. So now we will have more passive way of studying during this phase, and to the more active part. You will work in smaller groups. And also we will study and watch some videos. Okay, it's 10 for, so it's 70:41 your time, so we have enough time still. So perfect. After 19 minutes, I will ask you, or in 10 minutes, 19 minutes, I will ask you whether you want a break or no. Okay? So let's start. Let me upload my presentation now here. Okay, so nucleus. In cell nucleus or in biology textbooks, we can refer, or the nucleus is often referred to as the genetic library of the cell, or the cellular organizational center. However, these terms only make sense when we break down the nucleus into its individual components and later into molecules. We will then relate them to the function of the nucleus in protein synthesis and cell division. Afterward, it won't be difficult and we will understand why some cells do not have a nucleus and while others have two nuclei, why some nuclei are highly active while others show only minimal activity, and why nuclei occupy such a large space. Guys, do you hear some echo behind me or no? Yes? Okay, 1 second. There was just, I apologize, there was just the phone ringing. Okay, let me get back. Okay, so regarding nuclear structure. Okay, so here we have it. Nucleus is encapsulated in a double-layered membrane. So if we zoom in a little bit, we see outer membrane and inner membrane. Okay, here we are able to see that. Uh, Also here we would see on this picture the structure of pores. Um, so double layered membrane, and it has inner and outer membrane. Numerous pores are in the nuclear membrane, and here we have a structure of one. If you see on this picture, this is the outer membrane, and this is an inner membrane, and this is a pore. Um, Pores serve to transport mRNA into the cytoplasm. So mRNA is synthesized in the nucleus, and through the through the pores it gets into the cytoplasm. Okay? Um, yes. On its surface here, maybe you see also ribosomes can be attached. Okay, so as they are attached to rough endoplasmic reticulum, they can be attached also to nuclear membrane here. Mhm. Now let's get to the internal environment of nucleus. Um, the nucleoplasm can be called also as a karyoplasm, and it's so cytoplasm is a fluid filling the inside of a cell, and inside of the nucleus we call nucleoplasm or karyoplasm. Okay, nucleoplasm or karyoplasm. Nucleoplasm or karyoplasm contains all the DNA and synthesized RNA. So, guys, which other organelle contain DNA in the cell besides nucleus? We already discussed that. There is one other organelle which can contain DNA in the cell.
Mitochondria.
Perfect. Great. Only mitochondria and nucleus can have DNA inside them. Besides those two organelles, there is no DNA present in the cell, only in nucleus or in mitochondria. But most of it's here in the karyoplasm, in the nucleus. Um, So here in the karyoplasm, there is the DNA, synthesized RNA, some nuclear proteins, enzymes, nucleotides. We will describe that very shortly, ions and water. The nucleus has also its own skeleton. Okay, so to maintain its shape, to keep that shape, it has also its own skeleton inside. Um, Yes, and when we observe regarding the microscopic picture, the nucleus under a microscope microscope, we do not see any chromosomes, DNA molecules, or proteins. Now, that brings us here um, to this picture, the black and white picture. Here we have two nuclei next to each other. We can see only light, light sections and dark sections, okay, in both. Light and dark sections, why are they important? Light is transcriptionally active. We will get to transcription very shortly. Basically, it's active. So light sections of the nucleus are active, and the dark, here, 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 are inactive. Okay, I will now write how do we call those sections. Light section we call euchromatin. Dark sections we call heterochromatin. So it's basically do you see that, guys, now? Perfect. So this is the spelling of the names of those chromatins, okay? So on this picture, euchromatin or light sections are prevailing or dominating. So most of the nucleus is active in these cells, as well as here in both nucleus. So in this cell, most of the uh, chromatin or DNA is active. Now, specific region of chromatin is the nucleolus. In this picture, we are able to see it here, nucleolus, or here, if you zoom in where the arrow is, we see the cell, then we see the purple bubble, this is nucleus, and inside the dark dot, or more in darker purple color, the little dot is nucleolus. Basically like um, a nucleus in the nucleus, a smaller nucleus. We call it nucleolus. In this picture, we have it here, nucleoplasm. Here, sorry, nucleolus. Okay, this balloon, let's say. It's not physically separated from the rest of the nucleus. It only appears as a distinct structure in images due to staining techniques. What does it mean? In histology, in your during your first year, uh, you will have to know different types of stains or colors we use for samples. Basically, we have a tissue sample and you pour a color on it in order to distinguish the structures. This is the, the most common stain we use. Do you understand the word stain, guys? Stain? Stain? Yes? Okay. We call it hematoxylin eosin. It's not relevant for you now. It will be relevant in a year for you. Hematoxylin eosin is the most common stain we use in histology. And basically, students have a saying, unofficial saying, what the histology basically is. It's 50 shades of pink, the students say, because you have plenty shades of pink, because hematoxylin eosin is a pink stain or prevails in the pink shades of a color. Yes, and nucleolus, we have it here, the small dot, transcribes the genes for ribosomes. So basically produce rRNA. That brings us to RNA and DNA. So nucleolus, as we have seen here, have genes for rRNA or ribosomal RNA. Let me now draw or write three main RNAs you should be aware of. Okay, rRNA, ribosomal, T, tRNA, transfer. Oh, 1 second. Transfer and mRNA, messenger. Oh, what's going on here? Uh, do you see that, guys, now? Okay, so those three types of RNAs we have to already know. Okay, tRNA, RNA, and mRNA. Uh, those are just really the basic types of RNA. I will show you the list. I will show you the list of RNAs currently known. So let me show you. We have to know really only a few. So as you see, mRNA, RNA, and tRNA, transfer RNA. Only those three are relevant for us so far. As you see, there are much more RNAs. Okay? Basically, it's plenty of them. We have to know only three. So let me get back to that list. tRNA, rRNA, and mRNA. Let me check what else I have regarding this. Mhm. Okay, we will discuss a little bit more the euchromatin and heterochromatin, and then we will continue after the break. We will decide whether we want break or no. The DNA and RNA, respectively. So, guys, as we said, euchromatin is the light section. Euchromatin is light section and is active part of the nucleus, yeah, or of the DNA. If and heterochromatin is inactive. What does it mean? Different cells require different proteins, so they transcribe different DNA segments. What we see as euchromatin in one cell may be heterochromatin in another cell, and vice versa. What does it mean? Basically, if we draw a DNA, yes, these genes here, all of the cells have the same DNA, okay? All of the cells have same DNA. But this sequence or segment of DNA, these genes produce or are responsible for some liver proteins, call them, okay, in the liver protein. But those genes here would be responsible for some kidney proteins. So, what is meant by that? These green genes or this green sequence of DNA would be active in the liver and will produce proteins. So it will be euchromatin or it will be light under the microscope. But this section would be heterochromatin or dark in the liver. But it will be totally opposite in the kidneys because in the kidney tissue, these genes would be inactive in the kidney, so it will be heterochromatin or dark, and these genes here, which are responsible for kidney proteins, would be active, and those would be euchromatin in a kidney sample or kidney tissue. Okay? Uh, yes. So basically, yes. Mhm. However, something. Okay. Basically, this is everything so far regarding regarding euchromatin, heterochromatin, and the structure of the nucleus. Guys, do you understand so far what we covered? Is it understandable? Yes. Okay, so one more time. Euchromatin is an active part of the nucleus. Heterochromatin or chromatin is inactive part. Okay, we have, and all of both are here in the nucleoplasm with another structures like water, ions, some enzymes, and mRNA. We have two layers in the nucleus, inner and outer one. Inside the nucleus we call nucleoplasm or karyoplasm, and on the surface of nucleus there are pores which serve as a door or a gate to get mRNA, messenger RNA, outside to the cytoplasm, outside from a nucleus. And ribosomes can be also on its surface. Okay, guys, time's up. Do you want a break or no? Okay, so each of you want a break. Great. So see you in 10 minutes. Okay, and we will continue. Okay, guys, welcome back. So, um, let me continue where we ended. Okay, so DNA and RNA. So DNA and RNA molecules in the nucleus are chemically less diverse. Molecules in nucleus does not. Okay, I will clear this page. Does not produce ATP or proteins, or most of the metabolic pathways do not occur in the nucleus. Nucleic acids, as DNA and RNA are, comes from the word nucleus. The basic structural unit of these molecules is nucleotide. Nucleotide. Okay, this is one nucleotide. Nucleotide consists of nitrogenous base here, 5 carbon sugar, and 1 to 3 phosphate groups. DNA is an abbreviation of deoxyribonucleic acid. Deoxyribonucleic acid. If we remove this OH group, we got RNA. This is a ribo or ribonucleic acid. Okay, so there is only one difference between DNA and RNA, and the difference is this oxy part in the formula. As you see here, another difference about DNA and RNA is those here, adenine, guanine, thymine, cytosine, and uracil, are the nitrogenous bases. These 5, all of them, are the basic components of DNA or RNA. Only uracil, one second, uracil is present only in RNA. Okay, RNA, if uracil is present only in RNA. Okay, perfect. So here we are able to see, if we go here on the another picture, here we have a DNA. This is the structure of DNA. One strand of DNA is this one. And if we go more in a zoomed way, this is it. This is DNA. Phosphate group, 5 carbon sugar, or deoxyribose, and a nitrogenous base, adenine, guanine, cytosine, and thymine in this case. Regarding the history, DNA is unquestionably the longest polymer in the organism. Back in 1950s, the woman scientist Rosalind Franklin and later Watson and Crick confirmed its double helix structure. What is double helix structure? This is double helix structure. This is a double helix structure of DNA. Okay. Um, so these bond or it consists of two molecules connected by thousands of hydrogen bonds between the nitrogenous bases. So if we see, there is one strand, and here we have the color lines. Those are nitrogenous bases. Okay, so let me draw that. Oh, here, nitrogenous base. Bum, bum, bum. There are some, and another strand has another. Okay, it's not alpha helix, we have just two strands, but it's for simplification. And between here, we have a bonds we call hydrogen bonds. Okay, they are not so strong, but they are strong enough to keep the DNA together. But if we want to unzip uh, and separate two strands without any consequences, we can do that. We will get there very shortly why we need such a thing. Okay, so hydrogen bonds are between nitrogenous bases. One more thing regarding the difference between the terms which can occur very similar. Nucleoside and nucleotide. This is not the correct spelling. I will. Oh, you see in the room check the correct spelling. Nucleoside versus nucleotide. Open the room chat, please. And here we have a correct spelling. Okay, nucleoside versus nucleotide. As we said, nucleotide is a basic functional structure of of, um, of DNA and RNA. However, the main difference between nucleotide and nucleoside is this. Nucleotide is everything here. So, nitrogenous base, 5 carbon sugar, and a phosphate group. But nucleotide is without a phosphate group, okay? So it compares only a sugar and base. Sugar and base is nucleoside. Most of the time we will use nucleotides in our discussions, but you will have to know, you should know what nucleoside is. You will understand later or during medicine studies why are nucleosides important. So far it's not important for us, but please just remember there this is not the same thing. Nucleotide and nucleoside are not the same thing, okay? The difference is a phosphate group between them. Um, yes, so mhm. I guess we can go further now. So next part is about histones. What are the histones? You see maybe the icon here. We will watch short video regarding that. Histones are proteins that serve as spools around which DNA is wrapped. So these green circles or bubbles are histones. And as you see, DNA is wrapped around it. Okay, is wrapped around it. They play a role in organizing chromosomes and in conserving space within the nucleus. Basically, they are saving the space and they are trying to be as efficient as it is possible regarding the space. So they wrapped DNA around. This is just one unit we called a nucleosome. Nucleosome is a one histone with the DNA wrapped around. And as you see here, one nucleosome is just this. Okay, what we are able to see on this picture is just this little part on this more complex structure, nucleosome. And there is another one, another one, another one, another one. Okay, so nucleosomes are proteins which wrapped around themselves the DNA strands in order to save space and to be more efficient. Each cell, each one cell contains almost 2 meters of DNA in their nucleus, almost 2 meters. But they are able, based on their function and their unique structure, to this 2 meters long DNA strand put in the nucleus, which is just a few micrometers big. Okay, so it's like unbelievably effective way how to save space inside the nucleus. Okay. Um, okay, so I think we can now watch the video. Wait a second, I need to find it. Okay, let me upload this. We will watch one very short video regarding histones. Do you see the screen, guys? Yes, perfect.
In this animation, we'll see the remarkable
Also, are you able to hear the sound? Okay.
way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, guys, so let me get back to the presentation, and I would like to play the video one more time to show you where exactly the nucleosome was. Have you noticed the nucleosome part, or should we play it again? Uh, should we play it again? Just for that short period of the video, the nucleosome part, I would like to show you. No, no, okay, okay, okay. Yes, no. Okay, so most of you went for yes. Okay, let me play it. Just please check out what is the structure of nucleosome here. Okay, so we have a histones and we have a double-stranded DNA wrapped around, and then those nucleosomes are creating and stacking each on themselves to create to be even more efficient. Okay, even more efficient and to save even more space. So let me start. Let me play that one more time, just from till the beginning. From the beginning.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell.
Okay, guys, so this is just a regular double-stranded DNA. Okay, so no nucleosomes, no histones. This is just a DNA strand, double-stranded DNA.
The process starts with assembly of a nucleosome, which is formed when 8 separate histone
So those here are histones already. So histones are getting to be attached to DNA,
protein subunits
and you see how the DNA is getting wrapped around them. Okay, so it's curving. Okay, so it's not more linear, it's curving and getting wrapped around histones.
attached to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome.
So you see the we have a histone of 8 subunits, and the DNA is uh, wrapped double times around the one molecule of histone. So basically it wraps 2 times around and it goes away again. It's the nucleosome. Okay, and then it continues,
Multiple nucleosomes are coiled together, and these then
and these they are stacking together and be even more efficient, even more spacious friendly. So they uh, spread on really just the minimum space.
stack on top of each other. The end result is a fiber of packed nucleosomes
So they see how they are really very highly packed. So to be really like to try to be as less spacious as it is possible. Great. Okay, so that was basically that. Um, Let me get back to the. Oh, what wrong? What wrong? What wrong? No. Yes. Perfect. Um, Let me get back. Yes. So that was regarding histones. I will upload this video if you want it to watch it individually. I will upload it to the Google Classroom. Okay, guys. Now the active part of the session comes on the stage. You will work again in smaller groups, and one group will focus on transcription, one group will focus on translation, and one group will focus on replication. Okay? Um, I suggest, yes, we won't have enough time to cover everything, but we will start today and then we will continue tomorrow. Okay? So I think not the not each group will have enough time to cover their part, but we will start at least with one of those, and also we will watch some short visualization videos for that. So let me now start who is going to do what. Okay, let me put you into breakouts, and I also give you the pictures you can work with. Okay, so okay, um, I would like to ask how much time would you like to have? Let's ask you. A is 5 minutes, B 10, C 15. One more person. Okay, so you want 10 minutes. We can adjust that, but for at the beginning, we will discuss. You can have 10 minutes. Okay, so Aoi, Misato, and Nao, you are going to check and research transcription. Aoi, Misato, Nao, transcription. Satsuki, Shoko, and Sora, you are going to check translation. Satsuki, Shoko, and Sora, translation. Yuiko, Yuki, and Yuta, you are going to check replication. Yuiko, Yuki, and Yuta, replication. Guys, familiar so far? Can we start? Perfect. If there are no questions, we can start. Okay, group, so this is a picture for transcription. You can start with your research, okay? Perfect. Okay, group, so this is a picture for translation. You can start with your research, okay?
I cannot hear your voice clearly.
Now,
Yes, okay.
much better. Okay, you can tell
No,
me.
no, no, no. Could you say that again, please?
That does it work now?
Yes.
And it was also bad before.
Eh,
Uh, are you able or were you able to hear me properly even during a class, or was it problematic? Do you hear me now?
Yes.
Okay,
Yes.
so I will keep it like this. Okay, the microphone. Translation. This is a translation picture you can use, and you can start with the research. Okay.
Okay.
Okay,
Okay.
perfect. Okay, guys, sorry, I'm uploading replication picture. This is it. Okay, so this is a replication picture you can use, and you can start with the research. Okay, so what is it? What does it mean? What's happening there? Okay, perfect.
Yes.
Mona, are you here?
Yes.
Okay, so I will put you to the to one room. I can put you here, for example. Mona, you are going to do translation, okay?
Okay.
Mm.
Okay, guys, so I added Mona here because she was stuck in the main room, so please collaborate four of you together, okay?
Okay.
Perfect. Guys, would you like to have more time?
No.
No.
No?
We don't.
Okay,
Yeah, we don't have. We
okay,
don't need any more
okay.
time.
アミノアシドコネクティッド。
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes.
Okay, guys. So, um, can we start with the transcription, translation, and replication? What do you think? Yes? So let's start with transcription first. What have you found about transcription?
Mm, Transcription is the copying of the required base sequence in DNA to mRNA.
Mm-hmm.
That's all.
Okay. Can you maybe try to explain the picture?
Hmm?
Anyone from your group? Ali, Misato, and now. Have you checked the picture?
You mean this picture,
Yes,
number 19?
yes, this picture. Have you checked that?
Um, I don't understand the meaning of check, but yeah, we've seen
Oh,
this
check.
picture.
Yes. Have you saw this picture?
Yeah, yeah,
Okay.
of
Can you
course.
can you describe it maybe? What's going on?
Oh, okay. First, there's DNA
Can you use a pointer, please?
pointer.
Pointer or a cursor. If you up there in the white bar, there is a cursor button or pointer button. In order to see our pointer, now I'm using cruising with my pointer on the
Uh,
screen. You see? And there is a button with like a star next to it in the upper bar. Yes, perfect. I see that. Yes. Now we can see it. Now we can see it.
hmm?
Click one more time on it.
I can't see any bars on the top.
No?
No.
Then try to click on the right down corner. There is a pencil button
Down corner.
here, like when I'm cruising, so just a little bit right from it.
There's only plus or minus button.
And nothing else? Why?
Nothing else on my laptop.
Okay, maybe I don't know. You should be allowed on that. And here, is there a mouse button or cursor button?
Mouse button. Yeah, I can see your red button.
Yes. So can you click on this one?
Okay. Uh, Nothing happened.
Okay, so maybe just try to explain the transcription process on the picture. It will be very great. Whoever from your group, guys.
Oh, okay.
Hmm.
Actually, we didn't talk much about transcription. Just only the copying base sequence to the mRNA. So
Okay. Okay. Okay, so we will do that now. Okay, guys, transcription of DNA. Here we have a nice scheme of what's going on here. Um, DNA here is a double-stranded genetic information located in the nucleus. What's happening? Transcription is basically when you produce an messenger RNA. How to imagine, or the very nice analogy would be, now we can the take nucleus as a genetic library of the cell. Why? Imagine you want to cook a dinner for someone or a lunch for someone. So you go to library and you will try to find a very nice cookbook, okay? A book for cooking with the recipes, so the lists and the guidelines, basically how to cook something you haven't cooked before, or you want to cook from a cookbook. But you can't take this book with you from the library. So what you are doing, you just rewriting the recipe. Okay, so um, you take a piece of paper and copy the recipe. Okay, and that copy of recipe is our messenger RNA, basically. So this is what you are doing. You just copying the DNA. So DNA is like huge cookbook where are recipes, and if you want to cook something, like in our case proteins, you trans first thing you have to do, go to library or nucleus, copy the text, copy the cookbook, and have a recipe and create a recipe, and the recipe is basically the mRNA. So DNA can be located anywhere, cannot be located anywhere else except mitochondria. So DNAs are only here in our nucleus. So when the cell needs to cook a protein, firstly we have to transcribe the respective gene, or in our case, respective recipe, in genetic terms, gene, into mRNA, which is our copy, and this copying we call transcription. And the enzyme involved is RNA polymerase. RNA polymerase is an enzyme involved in transcription. Then, if we have a finished mRNA, it passes through the nuclear pores into the cytoplasm. And that's where the transcription ends and continues with translation. Let me play the short video, to understand more clearly what we are talking about. Do you see the video, guys? Perfect.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind.
Okay,
This is where the majority...
This is the first thing for us to remember. RNA polymerase. Okay, promoter and terminator are not yet relevant for us. Okay, but RNA polymerase is an enzyme which is involved in creating and synthesizing messenger RNA is important for us. RNA polymerase.
Of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the
what else we need to mention? Maybe here was it very nicely described. We also define the 5' end, as you see the number 5, and the 3' end on the DNA strands. Okay, and also the bases we mentioned before, adenine, guanine, thymine, cytosine, and uracil, have complementary basis pairs or partners. Adenine pairs only with thymine, as we are able to see here. So A pairs with T, and guanine pairs with C. There is a helper for that. How you can remember that? I will mention it right now. So adenine pairs with thymine, and guanine pairs with cytosine. How you remember it? If you see they are rounded, those letters G and C are rounded, so they are pairs, and adenine with thymine, they are pointed. Okay, they have no like rounds. Okay, so they are pointed, they are like strict. So this is how you can remember them, which are complementary with which one. So A pairs with T, G pairs with C. Okay, let me get back to the video. Oh yes, sorry, I need to put it. Here we ended, I guess. Yes.
open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding
If you remember, guys, at the first on the first hour of our sessions, we were comparing eukaryotic cells with prokaryotic cells. Now, in this video, they mentioned introns and extrons. One more time. I will play that.
coding sections called includes regions called exons that code for a protein, and non-coding sections called introns.
Introns are non-coding sections of DNA, and exons are coding proteins or genes, are coding or active as well in DNA. Such as things such as introns, we are able to see here, and they are non-coding sections of DNA, do not uh, they are not present in prokaryotic cells. Okay, so this is the introns we were discussing a week ago. I guess, if I'm not mistaken, something like a week ago, when we were comparing prokaryotic and eukaryotic cells. So introns are not present in prokaryotic cells DNA.
In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process
Okay, so the 5 cap and these things are not relevant for you. Okay, so far this is a higher level of transcription. This is a transcription for advanced students. Okay, we are still at the beginning, so we are introducing us to this process. Nevertheless, these informations can be helpful because you will have to know them.
is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 60.
Okay, so this is something we will discuss in translation part. Nevertheless, guys, is it so far understandable, or do you understand what's going on in transcription? Yes, more or less. We will ask yes, no. Do you think that you have at least some understanding of what's going on in transcription? One more person. Perfect. Okay, so all of you have an idea what's going on. Perfect. I guess, guys, we can end it here because we do not have enough time to go further. So transcription, we will end here. One more time. We have a DNA and we need to produce a proteins, okay? For any reason. Doesn't matter now. How do we do that? RNA polymerase comes to DNA and starts to produce and transcribe our recipe, our recipe or our copy of recipe from our huge cookbook, because we want to cook a protein. DNA is our huge library with many cookbooks. We copy the recipe and we transcribe it, and the outcome is messenger RNA. Okay, messenger RNA. So messenger RNA is produced in the nucleus and it passes through the pores. and gets to the cytoplasm, where the second step waits for us, and it's translation. Okay, so we will get there tomorrow. Um, I will upload the videos, and guys, just a short notice, the recordings from our sessions will be accessible till the end of this week. Okay, and I will also upload the presentations when we end the cell biology chapter, and we will move further with biology of man and genetics. So I will upload also the slides from the sessions that it will be accessible for you. Okay, doable? Perfect. Guys, I would like to ask about your feelings on the structure of the session, whether it was helpful for you, or would you like to maybe change something, or if it was okay. Thank you very much. Great, thank you. So we will stick with this structure. So at the beginning, we will have some, again, intro activities, and then we will move to the core. Tomorrow you can wait that or expect that we will move to the translation, and also we will move to the replication, and another topic we will focus on is cell cycle, basically. Okay. Any questions, guys? No? Okay, so thank you very much for your, again, activity, and see you tomorrow. And I will upload some videos we already saw today, okay? So you can watch them again if you want. Perfect. Thank you very much, guys, and see you tomorrow.